Thank you, Harsha. So bear with me, my voice is a bit raspy, so I've got enough water. Hopefully it'll last me throughout the whole talk. As Harsha mentioned, I'm going to talk about our work in patient-specific design and printing, 3D printing of pediatric devices today. Before I begin, though, I'd really like to acknowledge that this work is really the result of the efforts of a number of people, a number of clinical collaborators, Glenn Green, who I've worked with on the splint, and Dave Zoft on some of the data I'll present, Matt Wheeler, who we've done some large animal work with, Colleen Flanagan, who was my lab manager in Michigan, who did a lot of the laser sintering manufacturing that I'll show you here today, and the support of the NIH, especially for the tracheal splint. So what I'd like to do today is first talk about what is the paradigm of patient-specific devices. And basically, this is combining design based on patient image data with 3D biomaterial printing. And then in the rest of the talk, I'm going to give some applications that we've utilized it for, including creation of external devices, custom CPAP masks for kids with craniofacial anomalies, the splint to treat tracheobronchial Malaysia, which I'll refer to as TBM, so I won't have to say that during the whole talk. And then its potential use as a platform for regenerative medicine. But then I'm going to end with talking about, obviously, I think from a technological standpoint, this is a paradigm we can use to create patient-specific devices. But there's still a difficulty in how do we actually bring these devices into clinical use, and what is the business model to create around this technology. So when you look at some of the challenges in creating pediatric devices, one is obviously pediatric patients have a wide range of anatomy from very small to large kids. And with this variable anatomy goes variable function. I mean, just by the fact that pediatric patients weigh different amounts means they put different functional load bearing on their devices. And for both of these, that really requires that we can design for the individual patient, and we can use image data of that patient as a basis for design. The other thing that we don't really think about much in adults, obviously, is growth. And whenever, especially you implant a device, that device has to account for growth in some way. If it's a permanent device, the way you account for growth is you have to go back in surgically and remove the device and potentially put in a second device. And if possible, obviously, you'd like to avoid these surgeries. So we have to think about designing for growth, mechanically allowing growth, and the use of resorbable materials so that those implants will go away and you won't have to go in surgically and remove those. So there, again, we need the capability to do image-based design and simulation of growth even, as well as printing with 3D resorbable biomaterials. And something I think unique in some ways to the pediatric market that we don't see with, well, we do see with some issues in the adult market, but especially prevalent in the pediatric market, is the fact that you have, which is obviously good, you don't have large patient populations, but that makes it challenging from an economic perspective in terms of developing manufacturing for these small markets. Pediatric conditions are typically niche markets, and even within one market, you might have the need for variable design and different types of devices. So you have a very fragmented market, and it's very difficult to address this with traditional manufacturing because it's so expensive. If you're doing injection molding or something like that, it's very expensive to make molds. And if you have to make very many molds, I mean, you're talking months of time and hundreds of thousands or millions of dollars just to set up a line to do manufacturing. And that's where, again, 3D printing is its paradigm can really help us address this low volume market for pediatrics. So this is really the paradigm we see, and not just us, many people, of how to utilize image-based design with 3D biomaterial printing for patient-specific devices. Obviously, we start with a clinical need, and from that, we get patient data either through photos or scans or traditional imaging techniques like CT. And then we create a patient-specific model from that image data. And we can do this and also do simulation based on that model, come up with a design, and then fabricate that design using not just 3D printing or additive manufacturing, but combining that with other manufacturing modalities. And these are really the key technological developments that enable patient-specific devices. One is the development of software that we can utilize image data, because image data obviously is voxels and it's in a certain format. We have to be able to manipulate that and come out with a surface representation. And that surface representation, called STL, was actually made first for 3D printing, but now we utilize it quite a bit for design as well. That's one advance that's enabled patient-specific devices. The other one, of course, is 3D printing and the capability to print biomaterials that we can use for external devices or that we can implant actually. And if you look at the needs in terms of technological research, the ability to do very sophisticated simulation with these patient-specific models, fluid-solid interaction for cardiac and respiratory as well as broadening the range of biomaterials that we can print with. Those are the technological challenges we face in advancing this field. And then, of course, combining these patient-specific devices potentially with biologics for regenerative medicine. So this is the paradigm as we see it for patient-specific devices. This just shows you how we actually implement that, and we think about design from a multi-scale perspective, especially if we're thinking about regenerative medicine, because we have to make porous structures and we have to be able to embed biologics within the structure to do tissue regeneration. And somebody in architecture once said in a quote that the key to architecture is knowing where to put the holes. So we have to think about that too for scaffolds in regenerative medicine. We have to design layouts of materials to give us certain properties, combine that with our anatomic representation of design, add fixation, and come up with a final design. So we're trying to design for function on multiple scales. And then, of course, when we do this, we tend to end up with very complicated structures that, even if we had all the money in the world, would be difficult, if not impossible, to build using traditional manufacturing techniques. And that's why we have to turn to 3D printing. Now, even within 3D printing, there's a wide variety of technologies, from nozzle-based machines to laser-based machines. They each have their advantages and disadvantages. With the laser-based system as shown here, which we have used in the past, the nice thing is, because you use essentially this powder bed here as support, you can build a wide variety of very complicated porous structures. The difficulty, of course, is adapting materials to use in this system. You have to be able to mill these materials and create particles on a very small scale. And they have to have the thermal characteristics that you can use within the laser sintering. We can also use nozzle-based systems that use particulates or filaments as a basis. The nice thing about these machines, they're very flexible. You can use a wide variety of materials very easily in these machines, but they don't allow the complexity of building that we can get with these laser sintering systems. So again, we try to mix and match these technologies depending upon the goal that we're trying to achieve. But either way, this sort of image-based design, we create what's called an STL file. That feeds into any 3D printer. We can then build from a biomaterial and get the final product as shown here. So with the laser-based sintering process, you can see that we can build structures with a wide variety of complexity. The caveat being that these are all made from one biomaterial, polycaprolactone, which, like any material, has its pluses and minuses. So that's the really advantage of laser sintering. You can build these complex, random, or organized porous structures. We've implanted all of these except for the brain. We haven't done that yet. But this brain is actually from one of the doctors on the doctor's show, so he gave us an MR, and then we built his brain on the laser sintering machine. But this just shows you what's possible in terms of the complexity of structures that you can build by combining image-based design with 3D biomaterial printing. So how are we using this to treat pediatric cases? So one of them is the development of custom CPAP masks for kids with craniofacial anomalies. And this project actually arose because of the Splint project. The mother of this child had seen it on TV, and she went into Glenn Green's clinic and said, look, I saw you 3D printed this splint, can't you, the problem with their son, he couldn't get a CPAP, he has sleep apnea because of his condition, couldn't get a CPAP mask to fit. And she said, why can't you just 3D print a CPAP mask? So that started a whole nother project here of creating customized inserts for CPAP masks to fit kids with craniofacial anomalies. And because in other conditions, we of course take CT or MR scans, we didn't want to do that because of the expense and exposure to radiation for these kids. But it turns out now, of course, there are many devices you can buy on Amazon, handheld scanners or scanners for your iPad, that essentially you can scan this child, create an STL model of their face, and you can use that STL model as the basis for designing the insert. You can see actually drawing the insert interface on the child's STL model, extruding that from the face, getting an insert that can actually fit. We actually CT scan the mass shell, get an STL file, fit the insert into that, print a mold in this case, although now I think we can print with medical grade silicone, create the insert, and then we have a custom CPAP mask for this child. So they're currently running a clinical trial at the University of Michigan with a small number of patients, 5 patients, for kids with craniofacial anomalies to create custom CPAP masks. In terms of outcome, they're looking at leakage and compliance, which you can measure because if you have a CPAP mask, you know that they can measure compliance for the insurance company because they want to make sure you're using it. And they saw that with this custom insert in the same mask, they could reduce the leakage, the air leakage around the face on this patient from 25% to about 6%. So this is one application that actually probably does have a much larger market if you go into adults, but for kids with craniofacial anomalies, it's a way to apply patient-specific devices to improve outcomes. The other example, of course, is we're looking at patient-specific implantable devices, and this becomes obviously more challenging because of regulatory issues and finding a material we can print with that you can actually implant. And our first application for this was the case of tracheobronchial Malaysia. This is not a very prevalent situation. In kids, it occurs probably in about, our estimate is about 1,000 to 1,500 patients per year in the United States. It usually occurs because of vascular malformations that create, like the aortic ring here, that create abnormal mechanical pressure on the airway. We believe that because of this, the airway doesn't develop its normal morphology, especially the cartilage rings don't develop, and therefore you have what's called a malasic or very soft floppy airway. And in very extreme conditions, when the child exhales and creates a vacuum in the airway, it completely collapses the airway and they can't get air out of their lungs. This is a difficult problem to treat, and the treatment of choice so far is to do a tracheostomy on the child and put them on mechanical ventilation. Mechanical ventilation is not a great thing to be on, especially for long periods of time. Kids die on ventilation, they get pneumonias, it creates issues with lung growth and all these sorts of things. So it's not the greatest solution to the problem. So this was really my first collaboration with Glenn Green because he said, I'd like to teach, his specialty is airway reconstruction. He's a pediatric otolaryngologist. He said, I'd like to be able to tissue engineer a trachea. And I said, that's a pretty far out goal. can we start by, at least building a structure and a platform? And he said, actually, if you can do that and we can implant it, we can treat this condition called tracheobronchial Malaysia. But when you think about it, even though it seems simple in one sense, you've got to either put something inside the airway or outside of the airway to keep it open, there are a lot of issues you have to deal with. One is it better to put things in or outside the airway? In adults, usually put in a stent inside the airway to keep it open. In kids, that becomes a problem because as the airway grows, the stent migrates. You get erosion of the stent through the airway that causes all sorts of problems. So we decided to go with a splint, which is a device. You still have to do a surgery, but you implant it around the airway. And then we have to have a way to suture the airway into the splint so it doesn't collapse to allow patency. We also have to make this device customizable because every kid is different in terms of their defect length, their size, whether it's bilateral or not. Obviously, since we're implanting the device, it has to be biocompatible. And growth, we have to consider growth now because our hope is that the airway will grow. And that's one of the ways that kids sort of grow out of Malaysia. As the airway grows because of Poiseuille flow, the amount of pressure you need to get a volume of air through a larger airway decreases by the radius to the fourth power. So if you can keep the kids alive and their airway can grow, we believe that the Malaysia over time will go away essentially. So the question is, how do we design a splint to allow for growth? Well, one thing is that we make this out of resorbable material, polycaprolactone, and it actually resorbs over about 3 to 4 years, which we believe is a long enough time to allow airway growth. But we have to mechanically allow the airway to grow, so when it pushes on the splint during growth, the airway will actually open. And to the best of our ability, we tried to go through the literature and find out what is the pressure that growing airways will exert. You can imagine there's not much data out there in that. So we did our best in looking at rat studies and growth of airways to come up with a criteria for airway growth, a stiffness criterion, and we came up with this. And eventually we'd like to be able to simulate airway remodeling, but there's really a dearth of data on this. If we have Malaysia, and we relieve the pressure on the airway, do the cartilage rings then develop normally? This is one of the big unknowns, I think, in treating tracheomalacia. So all of these, from a regulatory standpoint, we use as design inputs for our device, and we try to design the device to fit these inputs and test it as well. So this is how we go about applying the paradigm again. So we're going to start from image data of the child. In this case, we have a CT scan with contrast. Well, we can see the lumen here. We generate a model of the lumen. And this is actually the first patient. You can see on exhalation that his left bronchi, there's a discontinuity. It actually exists, it's just that it's totally collapsed and you cannot see the lumen. We then generate a design, and the nice thing with this software, we can actually input the digital design of the device into the patient's model. So we can simulate basically how the device fits around the patient airway. The nice thing about this is really illustrated here with our second patient. He had bilateral Malaysia collapse of both bronchi. We basically sat down with the surgical team on Wednesday. We designed this splints on a Thursday, and we printed them on a Friday. So that's the other thing in emergencies. You have the capability to create devices de novo and actually make them in a couple days. It actually took longer to sterilize it and let it outgas than to actually design it and build it. As I mentioned, we can also do simulations, and this is something because when you build customized devices, obviously, do you have a question or okay? When you build customized devices, you don't necessarily know how they're going to perform, right? So at least before you test them, you'd like to go through a variety of designs, and simulate how they might affect the airway. So we're starting to implement that now. We can actually simulate, due to reduction of tracheal properties in this finite element simulation, how the airway collapses. And then, because of this file format. This is the STL format that we use to design the device. We use that file format not only for the 3D printing, but we use it as a basis for generating simulations of the device. We can implant the device, simulate the suturing of the device to the airway, and show that the device will maintain patency of the airway once the airway is sutured into the device. So we can now implement patient-based not only design, but simulations into creating devices. Of course, one of the things that the FDA was concerned about, as we were too, is you're putting this device around the airway. When it grows, are you going to restrict growth of the airway? So we wanted to look at different criteria for stiffness of the airway and see how they affected growth. We did this in a pig model. So essentially what we did is we sutured a splint around the trachea as shown here with different stiffnesses. And we could show from our original design input that if we did maintain stiffness less than 4 Newton per millimeter, as shown here, we could allow essentially normal growth of the porcine airway. Of course, one of the difficulties is that it's hard to engender true Malaysia in an animal model. This was an intact airway. We've done it by resecting cartilage rings in the animal model, but that is not really the etiology of the human condition. So creating an animal model that mimics this condition, I think, remains a challenge as well. And this was the first patient that we did, and something that I don't recommend doing is we actually did this patient as an emergency basis before we'd done anything in an animal model. So we had no idea if it would work or not. We had built some prototypes, but because of the emergency nature, the FDA gave emergency clearance for this patient. They likely would have died. They were going through respiratory arrest in the ICU. So they went in, and essentially his pulmonary artery was compressing his left bronchus. And the first thing the surgeons do, the cardiac surgeons, is they move the vascular structure to relieve pressure on the airway, but that doesn't create patency of the airway. So they made the decision to go ahead and put the splint on. And we actually build a model of each patient's airway for the surgeons to take in to try the different splints. And this is actual implantation of the splint on the left main stem bronchus of this patient. These are the first 3 patients that were done. As Harsha mentioned, we've now done 8 patients with a resorbable splint and 1 patient with a permanent splint. This just shows the digital model. Again, we're applying this paradigm of creating a model of the patient, creating the device design, implanting, making sure digitally that the models fit the airway, and this is the actual implantation, in this case the bilateral splint in the patients with the Malaysia. We actually published the first case in New England Journal of Medicine about almost 4 years ago now, and we did a follow-up of the first 3 patients in Science Translational Medicine, where the longest follow-up was about 4 years. So again, one of the things we're concerned about is, does the device restrict airway growth? So we're monitoring on CT scan the size of the lumen compared to the control untreated bronchus lumen. You can see this is the expiration CT scan of the first patient before surgery, complete collapse of the left mainstem bronchus. This is the CT scan immediate post-surgery. You can see that the bronchus is patent. This is tracking of the hydraulic diameter of both the right untreated bronchus in the red here and the left bronchus with the splint. And you can see that the treated bronchus basically grows at the same rate as the untreated bronchus. So based on both our large animal data and our patient data in 3 patients, Kaiba, the first patient, is now 5 years post-surgery, and he's doing well. So that was for kids. So in kids, we expect young infants, basically, who are typically less than 1 year old. We expect that if we can keep the airway patent for 2 years, that the airway will grow. We hope that at remodel, that's a big unknown, to have better mechanical properties. Although we know in Kaiba, the first patient, the airway the splint is now resorbed and he's doing fine. But in adults, we don't expect the airway to remodel or grow. So our hypothesis is that we need a permanent solution to keep the bronchus patent. In an ideal world, the way we would do that is we would put a splint in, we would deliver cells from that splint and sort of make exterior cartilage rings to stiffen the airway. We can't do that yet. So instead we use a permanent material, and this is PEC. So this is a girl who was 14 at the time of surgery. You can't see it here, but this is her model. Again, we're using the data to create a model and design the splint. You can see that this is the innominate artery, and it's compressing the airway, which is in green behind here, and it's creating Malaysia in the distal part of her trachea. So the red here is actually the design of the splint and the implantation in the digital model. You can see this is actually printed model of her airway. You can see the Malaysic segment right here. This is a splint in the OR and how the splint fits over the printed model. So the nice thing about 3D printing, of course, which people have done for a long time, is you can actually do surgical planning and procedure planning. You can sort of see and practice the surgery before you even do it. This is actually placement of the splint on the printed airway. This is a reconstruction of the CT scan before surgery. What you can see here is this is actually the trach tube she has in. You can't really see the lumen of the trachea because it's basically collapsed around the trach tube. This is her trachea post-surgery with the splint, and you can see that it has basically the full diameter. So this was Hannah, and this was, they had an article about her in Detroit News about 2 years ago, a year and a half ago. This is her pre-surgery. The doctors actually recommended to her parents to take her off ventilation. They didn't think she would live. And this is her 1 year post-surgery with her sister, older sister. So you can see that now conditions that, like I said, there are only probably 1,500 patients a year with severe enough Malaysia to treat using this technique. But nobody would have done that before because of market with 1,500 patients, no device company is going to build a device for that. It's just not economically feasible for them. But if you have this capability to take patient data, design new devices, and actually print them in an economically feasible manner, you can create solutions for these types of markets. And the thing we obviously want to go to next, and I know it's near and dear to people in this building, is regenerative medicine solutions. So try to use these 3D printed materials as platforms, obviously, for delivering biologics. And I'll just briefly show you some of those examples. Obviously, the advantage of this technique of using patient-specific data, designing complex structures, extending those to porous structures for scaffolds, very nice for complex structures like ear reconstruction or nose reconstruction. So in this case with Microtia, which generally is unilateral, affects only one side, you can actually take a mirror image of the good ear, generate the anatomic structure of that, design a porous structure from which you can deliver the biologics, create a porous anatomic design, 3D print that, and then in this case, we're using a hydrogel with about 30 million chondrocytes to seed the device. This is the ear actually implanted under the pig skin. Looks sort of like a normal ear. And it can show that you can get cartilage regeneration within this device. So this is really the next frontier, if you will, for patient-specific devices, is to use patients' own cells, seed those into the scaffold, deliver those, and try to regenerate tissue. Another application that we've looked at is for complex cranial facial reconstruction. So in this case, if you have a large tumor in the face, or you have to do resection, or you have trauma, the treatment of choice is to do a vascularized free fibular flap. So you take a piece of the fibula out with a vascular pedicle. Your surgeon hooks it up into the face, cuts it up, puts plates on it. You hope your surgeon is a very talented artist and can do that well in the operating room, although now you can plan it. The difficulty, of course, is that's a very difficult, long surgery. It takes about 15 to 18 hours to complete. Many people cannot undergo that surgery because of peripheral artery disease. So one idea is why not actually grow the reconstructive piece in the patient's back and vascularize it and then transplant it. So this has been attempted, believe it or not. It's been done probably on about 10 patients in Europe with varying degrees of success. One of the problems is obviously what you'd like to do is be able to design the reconstruction a priori, build a scaffold with associated fixation, print that scaffold, deliver the right biologics, whether they're cells or BMP2 or other combinations, implant it in the muscle for potentially a month to 2 months, and grow both bone and vascularity, because one of the biggest challenges, obviously, with large bone reconstruction is getting enough vascular supply. Grow the bone, as shown here, in the muscle. So this is, yellow is actually the scaffold. The blue spots here are the bone that's growing inside of the muscle. And then you can take the scaffold, the bone, if you need skin or muscle, with a vascular pedicle. You can hook that up in the face and hook the pedicle into the artery and vein in the neck. And then you have a perfused tissue construct with bone. And now you have, from the pedicle, because you've implanted this construct near the pedicle in the muscle, you have blood vessels growing through the construct, and you take those and you transplant those in the face to do the reconstructive surgery. Obviously, this is sort of far out there, but I think the technology exists really to do this now. And then finally, I'm going to talk about, we have attempted this in patients, not in the United States. This patient was done in Italy. And I would have to say it was not the optimal attempt, I believe. I think we have to have much better understanding of how to deliver biologics from these scaffolds, how efficiently we deliver biologics. But this was a case of periodontal disease. You can see this is you get resorption of the bone around the tooth structure here, as you can see here in the scan. This is very difficult to deal with. A lot of grafts, bone grafts in this case, fail. And in this case, this sort of shows the potential applicability of this paradigm. We get a scan from Italy of the patient. We design the scaffold. In this case, it's a two-phase scaffold because we want to attempt to grow a periodontal ligament, which we've done in rats, and a bone region, fit that scaffold onto the tooth defect. We print actually a phantom of the patient's model and the scaffold, and we show that we can get good fit of the scaffold onto the phantom. And in this case, we were just delivering PDGF, or platelet-derived growth factor, from the scaffold. I think we've had much better success in the rat model of delivering periodontal ligament cells in the PDL region, and then transduced gingival fibroblasts to make bone in this outer region. But obviously, those are difficult from a regulatory standpoint to get into clinical use. And this shows the initial scaffold. So we built this scaffold at Michigan. We had it sterilized and shipped it to Milan, Italy. This is actually the implantation. You can see the scaffold here into the defect around the tooth root. It did pretty well, actually, for about a year. This is 6 months post-op. This is a year post-op. It looked good. But then one of the difficulties is with these sorts of procedures, and also the difficulty with bone graft, is we have dehiscence of the soft tissue around the defect. So you can see actually the scaffold coming out here, the mucosa here as dehisced, and basically the scaffold had to be removed. So that's really the challenge, I think, in biologics, to avoid dehiscence. Number one, we're looking at trying to create gradients of going from soft to hard tissue, or maybe multiple tissues, to reduce mechanical stress on the tissue. And also really growing the tissue having robust tissue growth underneath where the soft tissue is important to avoid dehiscence. So these are some of the challenges that remain in using these scaffolds for ear reconstruction, nose reconstruction, as well as reconstruction in the mouth is dehiscence. So I've showed you some ways that we can apply this paradigm to treat patients. External devices, which are probably the lowest hurdle of getting into regulatory approval. Implantable devices, which we've had success with and are now going into clinical trial. And then eventually going out to combine printed scaffolds with biologics for regenerative medicine. So definitely there's a technological paradigm there we can follow to create devices. But is there a model to do this? Is there a path? And what are the challenges that we face? So one of the challenges, obviously, and it's a good thing, we don't have a lot of kids that have these conditions, but those that do sometimes face very dire circumstances. It's a very small market, and this is a challenge for most manufacturing technologies. But if we combine patient-specific design with 3D printing, we can address these small markets. And that's really one of the fortes of 3D printing. We can make, there's sort of the old cliche that complexity is free and customization is free with 3D printing, and that definitely applies for pediatrics. I think you've seen that we do really require patient-specific devices in pediatrics because there's wide variations in anatomy and function. We have to be able to design for growth and low-volume markets. I think image-based 3D design and 3D printing allow us to do this, to address these challenges, and especially to address the challenges of low volume, the need for low volume, highly customizable and highly complex devices that could be extended for regenerative medicine. But there are challenges as well. I think even in these cases, companies still find it challenging to develop these devices because of the low market size, and their really goal is not only to develop devices and treat patients, but of course they have to make profits to keep existence. So I think there may be a sort of a dead zone, maybe a no man's land, I don't know how to really phrase it, between companies developing devices for large markets and academics trying to develop markets or develop devices for these markets. I think more and more you'll see hybrids between the two because of the capability. Like before, with a group of 3 people, we never would have been able to set up the manufacturing capabilities to create these devices. But now, because of additive manufacturing, they have the capability to do this. But academics, I think, is still a difficult venue in which to develop these devices because of the really focus of academics. There are different cost and reward structures that make it difficult to really pursue regulatory issues and translational funding in academics, because we all know that publishing in Science and Nature, it's really about the first discovery, and doing fatigue testing for devices and degradation testing is not something that will get you published necessarily in these high-profile journals, but if you're trying to get something through regulatory approval, it's absolutely required by the FDA. So I think those are some of the challenges that we're facing, plus the fact, and this is a nice graphic from Tony Radcliffe, the regulatory cost to get something into market, and this is just our own data from the splint. We estimate it'll probably be in the neighborhood of about $6 to $7 million total cost with preclinical testing and the clinical trial to get it into the market. So what is the future? I think if we can take this paradigm and really develop it, the 3D printing, use a few technologies, 3D printing technologies, but adapt them to a wide range of biomaterials. Provide robust and really flexible and rapid patient-specific design and simulation, and rely a little bit more on computational analysis to prove out devices. And even the FDA is looking into this paradigm more and more. If we can put the design and the manufacturing, flexible manufacturing, in a quality environment, a GMP environment, and prove, for example, biocompatibility for a given material, for a given process, and have that standardized, that will at least allow us to reduce some of the regulatory costs and time expense of getting devices into the market. And actually, this is what we're trying to do now, and that's one of the reasons I was excited to come to Georgia Tech, is Georgia Tech was really willing to try to help put this together, and that's what we're doing now with GCMI. We have a couple machines, a laser sintering system and a bioplotter that we're going to put in a clean room and build around GMP. So our hope is that this will be a plug-and-play environment, that physicians can have ideas for devices. We can work with them to design these, to simulate them, and then we can actually build them rapidly in this quality environment. And that's really our goal, at least, for our hope to try to address some of these issues. And with that, I've talked about the paradigm, how it's really enabled the development of pediatric devices, the technical challenges of developing patient-specific simulations of devices, expanding the range of 3D printable biomaterials, but also how do we create a business model around this to get these devices into clinical translation. And with that, I'll end and be happy to answer any questions. Thank you.
Scott, first of all, congratulations. That's an amazing story that I've heard several times.
I know, I've probably seen this pretty much the same talk. So.
And I enjoy hearing it every time. My question is, one of the unique things that you do is you don't just 3D print. You do the image-based computational design at the same time. And my question is, do you think there'll be a point at which the FDA, which currently approves a design, will accept computational data as validation for a patient-specific design?
Yeah, it's a really good question. I mean, I think they are definitely moving into that, to that point where they will. I know I've heard just anecdotal stories where if at least in a revision of a 510K where a company had a device and they were changing something, it was a flow device, and to change it, the amount of testing they would have had to do would have been enormous. And the FDA really encouraged them to do a lot of CFD simulation and try to prove their design would work for that, and they accepted that as a subsequent 510K approval. I think they will move towards that. Of course, they will want significant validation of the models. There are guidance documents out there on using computational simulation as part of the submission process. I think with these types of devices, you're going to have computational simulation as a component of the regulatory approval. I don't think it will ever replace bench testing and animal preclinical testing, but it's going to play more of a significant role in establishing your bounds of your device, your worst case scenarios that you will then test before you bring it to market. Yep.
A couple dozen questions, but I'll only ask one now.
Okay.
You didn't mention immune response or inflammatory response in any of these. So the rookie question is that not a concern, or how do you think about that, and how do you handle it?
No, I think it definitely is a concern. We haven't seen, at least in the patients, at least clinically, a significant immune response to these devices. PCL is a fairly well-known biomaterial in terms of its biocompatibility, and we don't have a very great way to track immune response in the patients, but at least anecdotally from clinical follow-up, we don't see any problems, and obviously we know that the device is degraded and gone away, but that's something that the FDA obviously is interested in, the immune response, and when you get breakdown of these materials, so we're doing an animal study. We're hoping to launch that in about a year or so to study these issues. But it is a key question, and just my own lack of expertise in immune response and things like that, probably not focused on as much as we should have. Yep.
Yeah, with the way this process works right now, where's the liability involved? All on clinicians, or are you viewed as a second opinion? How's that?
So I think right now, probably most of the liability, which is probably makes the institution nervous falls on the institution and somewhat on the physicians. We do, of course, even these emergency cases, they all go through they all went through IRB at the University of Michigan, and we deal with the FDA, and they have a specific procedure to do emergency clearance. And of course, you go through all informed consent with the parents. But I think in the long run, still, the liability rests on the institution. But again, I'm not a legal expert, so. Yep.
Speak to the question of like yield repeatability, in the sense that in other disciplines, like the environment in which you're designing the materials you're using, they can have like wildly different variations. And could you speak to the proportionality of what percentage of these products are done actually at the hospital, and how expensive it is for the hospital to create that environment? Or is it just primarily just outsourced at one contractor that everyone knows is really, really good, and they just deal with that?
Yeah, that's a really good and important question in terms of the variation. 3D printing, I think, compared to a lot of manufacturing technologies, gives you a little bit more variability in the outcome. So when the FDA looks at processes, they either want you to validate them to show that every time you give the same input, you get the same output. I think that's difficult to do with 3D printing. There are actually variations in terms of just with the laser sintering process, the position in the build platform in which you build the device creates some variability in the device. As well as you mentioned, the material, the powder, humidity in the room temperature, these all affect the reproducibility of the device. So I think, and the FDA has actually issued a draft guidance on additive manufacturing, but the way we do it is that we actually build coupons with each build at different locations, and we can verify the geometry of the device by micro CT to see how it compares to our design geometry, and then we do mechanical testing of those coupons to see the variability compared to what we expect. So in that sense, we really do verification of it. In terms of devices, to my knowledge, no hospital is building implantable devices on site. They are doing surgical model planning, which has some validation issues, but none to the degree of implantable devices. We have done that at the university to the best of our capability when I was at Michigan to build these devices. Obviously, that's one of the reasons why we want to implement it in a quality environment and a quality system to get to track reproducibility as best that we can. In most cases, implantable devices right now are being built by some companies, and they're going through the same issues we are in terms of quality system and quality implementation. But it is a big challenge.
Great talk.
Thank you.
You talked about kind of like going through the regulatory process. Do you have some sort of concern that this technology is going to get like a non-coverage determination from CMS, which would then kind of exclude the pediatric population on the chip, like low-income families in those cases, which would be kind of the more challenging in terms of cranial facial abnormalities?
I don't think necessarily that CMS would not reimburse because of the process. I think it would depend upon, what is the condition and what are the clinical alternatives. As long as you can prove that what you build with the process meets your quality standards, that meet your design inputs, and the FDA, you have to, obviously, if it's like the splint would be a humane use device, so it'll go through an HDE. And in reality, if you can do the splinting, both from a quality of life and outcome standpoint compared to ventilation, which is the clinical gold standard, as well as even a cost standpoint, I mean, if you put a kid on a ventilator for 2 years, we looked at our own numbers, it'll run between $750,000 to $1 million, not to mention morbidities associated with ventilation. So I think from that perspective, if you can prove, if you get it through the FDA, you do the trial, you prove it's a better treatment, and the fact that, the cost, a lot of these kids have, cardiac anomalies that have to be treated, so the surgery might run $200,000 to $300,000. You still have a significant cost savings and a better outcome. So I think from that perspective, you would expect CMS to okay it for reimbursement. I think the demonstration of quality and reproducibly is really going to come through the regulatory process. And if you can do that and you're a better treatment for the condition, I think it will be reimbursed. But again, I'm not a reimbursement specialist either. Yep.
Foresee any improvements in 3D printers themselves, or expect any improvements that could affect your product?
Yeah, I think there will be a lot of improvements, especially, I think on the nozzle-based system side, you've seen tons of companies now developing new systems. A lot of people are looking at adapting new biomaterials to these systems. I think the nozzle-based systems are easier, it's an easier technology to which you can adapt new materials. So I think you'll see, and if you look at the literature, you'll see people developing new printable materials all the time. I think the laser sintering is a little bit harder because you have to be able to mill and process the material. It took us a long time to find somebody who could mill PCL because it's very ductile. If you use mechanical means, you sort of squish it. So you have to use liquid nitrogen jet milling on it. Those things probably hold, you need technological development not only in the 3D printers, but in the processing techniques you use on the materials to prepare them for 3D printing. So I think that's as much of a bottleneck as the 3D printers.
If there are no other questions, I'd like to welcome you to thank Dr. Hollister once again for his talk.
Thank you.